And welcome to Introduction to Psychology. Today we are going to be talking about neuroscience, and we're going to talk about the different aspects associated with neuroscience in terms of the history of neuroscience, and also talking about the basic foundational elements associated with the field of psychology from a neuroscientific perspective. So let's begin first by talking about a brief history of the field of neuroscience and where it is that we began this idea of trying to understand where the biological components really stem from. The reason that this is so important to us is because the first questions that we began to ask that were very philosophical in nature were related to the mind and talking about where is the mind. And a lot of these issues related to trying to identify the location of the mind by saying, was the mind in the head? Was our mind in our heart? And if you really look at statements made by individuals across many centuries, you can actually try to understand how it is that people would assume that the heart, being such an important mechanism within our bodies, and it dictates so much for us in terms of our ability to be alive, we would use this information to being able to decide that that's potentially where our mind or where our soul might potentially be. That's why we developed ideas such as follow your heart or listen to your heart as being the heart, the seat of the soul, or where our mind really is. Now, as we've been progressing through our field in terms of it becoming much more scientific over centuries, we've obviously been able to make leaps and bounds in terms of trying to identify where it is that the mind would be or where we think that the mind is. This is why we now tend to focus so much on other areas, specifically in the brain, and trying to identify the brain as being that area of where it is that the mind is located and what it is that the mind can do. So when we think about trying to understand how the brain has really developed into its current form of why it is that we care so much about the brain and why it is that we try to understand the brain, we've been really trying to figure out the brain for a very long time. Aristotle was one of the first individuals who really tried to understand this idea of the heart and also of the brain, and looked at this idea that potentially mental functions were actually located in the heart. So again, going back to some of those statements of thinking with your heart, follow your heart, were ideas that were already stemming from the times of our philosophers. Now, fast forward here to about the 1500s, and Rene Descartes comes along and starts talking about the importance of the brain and starts talking about the importance related to reflexes being a part of brain functioning, and also trying to understand this idea again of the seat of the soul and this interaction between the mind and the body. Now, when you look at the work related to Rene Descartes, a lot of the issues related to Rene Descartes was this aspect of trying to identify in the brain where the seat of the soul would be. And the seat of the soul, according to Descartes, was located in the pineal gland. Now, where is the pineal gland in our brain, or why is it so unique, and why did he care so much about it? Well, the pineal gland was so important for Rene Descartes because he identified in individuals that the pineal gland was actually very unique. There was no other mechanism or part of the brain that looked like it, or there wasn't other ones within the brain, right? So there was no left half of the pineal gland and the right half of the pineal gland, but rather it was solitary. So thus giving him the idea that potentially the seat of the soul would be located in the pineal gland. Now, as I mentioned, through our scientific experimentations for many centuries, we've now been able to identify that the pineal gland is not the seat of the soul, but rather it's very important for regulations associated with hunger and also with sleep. So the idea, though, was that we were now beginning to give the brain its importance related to trying to understand its functioning in terms of this idea of the mind, right? That we're able to process information, and that we have functionality of the brain in terms of being able to do mental processes. Now, this also then leads us to trying to understand other areas of, we would say, pseudoscience, as being mechanisms that also were giving the brain and the head the importance that it deserved. Now, one of those things is the area of what we call trepanation. Now, the idea of trepanation was that individuals would make holes in an individual's skull, and the reason that they would do that was because they felt that by doing that with these types of tools here that are on your right hand side, we would see that these evil spirits would leave the brain. Now, again, these were from indigenous cultures that would look at this idea and say that these individuals were potentially possessed by evil spirits. But yet, in reality, what these individuals more than likely had was they probably had some aspect of a psychological disorder, potentially like depression or mania or schizophrenia, where these individuals would display abnormal behaviors in comparison to other individuals within those areas, within those cities or towns or cultures, and thus then say that they were probably possessed by evil spirits. And to allow those evil spirits to be able to get out, they would make these holes in the head here, as we see on our left-hand side, where these individuals now would then be able to allow those evil spirits to get out and being able to have that relief, right? Now be able to change their behaviors. Now, in modern times, right, and when I say modern times, we're still talking about 1700s and 1800s in terms of trying to understand more of the idea of how the brain worked, trepanation was still used, right? It was still used, but now for the ideas to increase blood flow to the brain and thus potentially creating greater intelligence. And they would do that through tools like we see here on our right-hand side that ultimately would then be used to release the pressure in the skull to be able to increase that greater intelligence for individuals. Now, as I mentioned, these were much more based on pseudoscience than anything else. We didn't really have, and we still don't have in modern times, any type of scientific evidence that would suggest that any of these types of techniques would actually be useful in terms of being able to create greater intelligence or be able to resolve any type of psychological issues, but rather we saw that they were much more founded in this idea of pseudoscience. Now, as I mentioned, our idea of the brain and the importance of the brain has been coming along for a very long time. One of the most prominent individuals who kind of gave us the idea of localization of functioning was Franz Joseph Gall, who developed back in the 1800s this idea of what he called phrenology. Now, what was phrenology? Phrenology was a representation of localization of functioning. So in other words, in different areas of the brain, we would essentially have different functioning associated with our mental faculties. So in other words, on the top right-hand corner of the brain, we see that individuals have a little bump on the head. And through that bump, we would ultimately see the psychological attributes being represented. So if an individual had a more prominent bump on the head than another individual, that would show much more of that attribute than others. So for instance, they would rub a person's skull and they would say, okay, I feel this little bump here that you have on the top of the head on the right-hand side towards the front. I think that you are a highly motivated individual based on that bump. And based on the prominence of that bump, right? So the more pronounced that bump was, the individual then would be said to have much more of that mental faculty or much more of that psychological attribute than other individuals. Now, phrenology was a very, very prominent idea back in the 1800s. It was so widely used and believed that individuals would actually use phrenological reports to actually be able to get married through the church. So what you would do is you would go to the church because you were going to get married to your partner, and they would ask you to do these phrenological reports. You would do your phrenological assessment, and it would be read to the individuals at the church, and then they would be able to decide, yes, these individuals are compatible or they are not compatible. Politicians would also use phrenological reports, because what politicians would do is they would say, I've done my phrenological report and look at these areas associated with honesty, with motivation, with charisma, with care, and so on and so forth, that ultimately represent that I am a good individual and I am going to do what's best for you, versus other candidates who don't have those similar attributes. So ultimately, using phrenological reports was a very, very easy way for individuals to convince others of the work that they were doing in terms of how it is that they were being represented in terms of psychological attributes. Now, other famous cases that we have associated with trying to understand the functionality of the brain was the most famous case that we have of who we call Phineas Gage. Now, Phineas Gage was a foreman when the railroads were being laid in the West. And unfortunately, he had an accident while working on that railroad where there was an explosion of a rock because he was using what's called a tamping iron to actually tamp down the charges into the rock. So what these individuals would do is they would get explosives, they would make these holes in the rock, then they would tap down these explosives using a tapping iron down into the rock, and then they would detonate the explosive and break those rocks apart. Well, in one of those cases, he was actually tapping down that charge, and unfortunately, there was a freak explosion, and that tapping iron went right through Phineas Gage's head, and that tapping iron landed 80 feet away from him. And as you can see here on the diagram that we have here on the left-hand side of this image representation of what it would look like, we saw here that it essentially went under his eye socket and straight out through his frontal lobe, right through the top of his head. Now, why is this case so important to us? It was so important because whenever it is that we actually tried to understand what happened to Phineas, we saw that obviously he had damage to the frontal lobe of the brain. Now, why was that important for us? Well, because it demonstrated to us from a psychological perspective and a biological perspective why the brain was so important for us, and specifically the frontal lobe. Because in the case of Phineas Gage had damage to his frontal lobe because of that tapping iron, and people began to report that they actually saw differences in Phineas before and after his accident. Individuals would report that prior to his accident, he was a very, very calm individual, was very understanding of others, never cursed, was very charismatic with others, and always sought to help others. After the accident, it was said that Phineas's personality changed, that now all of a sudden he began to use curse words, or that he began to become very aggressive with individuals, and he would yell and have outbursts of anger all of a sudden. So it was said that because of the damage that he received to the frontal lobe, it was ultimately found, or it was perceived, that now Phineas, unfortunately, had suffered personality changes, right? And we would say here that because of that damage to the frontal lobe, it changed his personality. Now, ultimately, when looking at this particular case of Phineas Gage, did his personality completely change? Well, no, potentially not, but rather some of his behaviors began to change, and we're going to talk a little bit more later on in lecture why the frontal lobe is so important for us. Now let's switch our attention to talking a little bit about the brain. So when we're thinking about the brain, there's a lot of information that we got to process in terms of how the brain works and what actually is the anatomy of the brain. So let's first begin by talking about the anatomy of the brain. So when we think about the brain, there's two major things that you always have to consider whenever it is that you're talking about the brain. And those two areas to consider, those two things that you have to consider in terms of that basic anatomy, are what we call the gyri and the sulci. So what is the gyri? The gyri are the wrinkles or the gray matter which contain neurons in our brain. Now, ultimately, whenever it is that we see a species have more gyri, this ultimately means that they are going to have a more sophisticated brain. What is that going to reference? That is going to reference their ability to do more complex processing of information. So ultimately, whenever it is that you see the brain of a species that has a lot of gyri versus sulci, ultimately that species, that animal, is going to be able to have more sophistication in terms of their ability to process information. Now, what is the sulci? The sulci is the spaces. So if we look back here at our brain and looking at our human brain, we see that we have quite a bit of gyri, right? We have a lot of those wrinkles or those folds in the brain, and we don't really have that much space, right? We don't have those empty spaces in terms of what it is that we're looking at in terms of our brain. Versus the brain, for instance, of a mouse, that you can see doesn't have a lot of wrinkles to it. It actually has more of those spaces, more of that sulci, versus that of a cat. So if you look at the cat's brain, you'll see here that it's got more folds, more wrinkles than in comparison to this mouse. So as we're looking at the species of the mouse, you'll notice here that there's one area of the brain that has a lot of wrinkles, a lot of those folds. And you'll notice that here where we see the occipital lobe, towards the back of the brain, where we see actually that brain stem coming out, right, towards the back. So if you look at that part right there where you see the occipital lobe has a lot of this gyri, a lot of these folds. Why is it that in a cat, right, in a feline species, we would have a lot of gyri, a lot of those folds in the occipital lobe? Well, think about vision for cats. They typically tend to hunt at night, and they're going to be great hunters, and they have great eyesight. So ultimately here, having very good vision is going to be correlated, right? It's going to be related with a lot of this folds, a lot of this gyri that we see in comparison to that of a mouse. That again, we don't see that much gyri. We have a lot more sulci in our mouse in comparison to our cat. Now, other basic anatomy that we need to concern ourselves with in terms of the brain is we also have to look at what's called the cerebral cortex. Now, what is the cerebral cortex? The cerebral cortex is that layer of tissue which covers the two hemispheres of the brain. And then, as I mentioned, we have these hemispheres in the brain. We actually have two hemispheres in the brain. What do we call these two hemispheres? We call them the right and the left hemisphere. Now, in terms of the right hemisphere, what do we find that is related to processing abilities of the right hemisphere? We tend to find that there's more muscle control on the left side of the body than we do on the right side of the body. We also find that individuals that are right hemisphere dominant tend to have better spatial abilities, better facial recognition, visual imagery, and music processing abilities, versus those individuals that are left hemisphere dominant, that we typically tend to see them having better language skill dominant, better math, and logic skills as well. Now, when we look at the brain, there's an important component here that we have to look at in terms of how our left and our right hemispheres communicate with each other. And the way that they communicate with each other is through what we call the corpus callosum. Now, what is the corpus callosum? The corpus callosum is this giant band of fibers that allows communication between the two hemispheres. So you can see that right here in this image that we have to the right, that you have that band of fibers that's connecting both the left and the right hemisphere in this image. Now, what we find here is that whenever it is that individuals' corpus callosums are severed, which does happen or used to happen more as a treatment many years ago, as a treatment for severe epilepsy, whenever it is that individuals would have severe epilepsy, the cutting of the corpus callosum was actually used as a treatment because it would stop that communication and that interference between the right and the left hemisphere that we would find. Now, as we do have connections between the left and the right hemisphere, and we do have control for the right hemisphere controlling muscles on the left side of the body, and the left hemisphere primarily controlling muscles on the right side of the body, this is ultimately going to be referring to what we call contralateral and ipsilateral connections. Now, what are contralateral and ipsilateral connections? If we specifically look at something like our eyes and the way that our optic nerves are connected to our occipital lobe, that area of the brain that processes visual imagery, we actually find that we have both contralateral and ipsilateral connections. Now, what are contralateral connections? This ultimately means that projections that we find on the opposite side of the brain are going to be controlled by that opposite eye, right? So in other words, objects that are going to be in the left field of vision can be processed by the right eye. Right? And we're going to process that information on the right side and on the right and on the left hemisphere as well, because we have both contralateral and ipsilateral connections. So the same thing goes for the right visual field. We have ipsilateral connections that are going to be processing of information on the same side of the brain, and also contralateral connections, processing of information on the opposite side of the brain as well. Now, as we were mentioning and talking about the two hemispheres communicating with each other and having that band of fibers in between those two hemispheres communicating with each other, we also have to recognize that each hemisphere independently can also communicate with itself. So how is it that the hemispheres communicate? This is where we're going to now introduce this term of what we call a neuron. Now, what is a neuron? The neuron is going to be this mechanism that information is going to be traveling to different parts of the brain for interpretation and bringing that information back to the appropriate part of the body for movement, for processing of information, for continued location or processing of auditory information, taste, smell, whatever it may be. We are going to have information being communicated from one point to the next, and we're going to do this through our neuron. Now, our neuron is made up of 4 parts. Those 4 parts being, number 1, our dendrite, which our dendrite receives information from other neurons. The second part is what we call our axon or our out. This is the part of the neuron that transmits information from the cell body to other neurons. Now, very important, our axons are covered in what we call a myelin sheath. Now, what is the myelin sheath? This is fatty insulation, because as we're going to be sending electrical impulses, because that's what neurons are going to be transmitting the information as, electrical impulses, that electrical information, we need to make sure it's insulated. And why? So that way that energy doesn't dissipate. So just like your home that has insulation in the walls and in the ceiling, why do we do that? So we can keep the cold air in, we can keep the hot air in whenever it is that we need that as well. Ultimately, we're going to use it as insulation and keeping that energy in. So that myelin sheath protects that axon and keeps that it functions as insulation and keeps that energy there in terms of that electrical energy that is being transmitted. Now, our third component is going to be what we call our terminal button or our terminal buttons. These are the end of the axon which release chemicals into the synapse, right? And we're going to talk about neurotransmitters in just a couple of minutes here. And then last, we have what's called our synapse, which are the gaps between adjacent neurons. So between one neuron and the next, they're not stuck together, but rather they're very, very small gaps that exist between them. Then those gaps, we call that synapse, right? Or the synaptic gap. And that gap, what happens for one neuron to communicate with the other, we have a release of neurotransmitters occurring at the terminal button. Once those chemicals are released, that dendrite, the one that receives the information at the next neuron, is going to be capturing that information at that receptor site and telling that next neuron to continue firing and doing and processing information as needed. So when we think about neurons and we think about how many neurons we have in the brain, we estimate that we contain somewhere between 10 to 100 billion neurons in our brain. Now, this is important for us because if we only had 1 or 2 or maybe 100 neurons in our brain, well, think about what that would mean in terms of neural damage, right? Like, let's say, for instance, you were in a car accident and you had brain damage. Well, if you only had, let's say, 10 neurons and you were in an accident, the likelihood that you're going to have a lot of brain damage is going to be pretty high. And you're going to have no communication between your perceptual senses, like your eyes, your nose, your ears, and so on and so forth. You're not going to be able to process that information from that receptor sensory system to the brain to process that information if you only have several neurons. But rather, if you have billions of neurons, that's going to be good for us because even as time passes and we may have neurons dying or we may have injuries, we still have other neurons that are still going to be able to complete the task. Now, this doesn't mean that if you're in a car accident and you have brain damage, the other neurons are all going to pick up the slack and you're going to be perfectly fine. Well, no, that doesn't happen because different parts of the brain are ultimately going to be responsible for different functioning, and ultimately they're going to need communication. And if you have major injuries, let's say for instance in a car wreck, right, where you've hit your head very hard, or you have a concussion, or you have a stroke, or some other event that may have happened that has created damage in the brain, you may ultimately suffer from some type of disorder in terms of being able to cognitively process information. Now, as we're talking about the neuron, it's important for us to understand what neurons look like. Now, as I was mentioning, our neurons are made up of these 4 parts, which again are going to be our dendrite, our axon, our terminal button, And then also our synapse or our synaptic gap that are going to be at the end of your neuron, right? Now, as you notice here, our neurons are also made up of our cell bodies. What is the purpose of our cell body? The purpose of the cell body is to create now what we're going to call an action potential. What is an action potential? An action potential occurs whenever it is that that neuron is told to fire. So once it is that that terminal button releases the neurotransmitter at the end of one neuron to the next neuron, if that neuron, that next one, captures those neurotransmitters from the previous neuron and it's told to continue firing, now the cell body will convert that information and will convert that energy from a resting potential state to an action potential state and activate the nerve, right? Activate that neuron and send that information through to continue that processing of information. Now, this is all happening very, very quickly. And we do have different neurons for different things. So what are the neurons that we typically tend to see? Well, our nervous system actually has three types of neurons. We call these the sensory neurons, which transmit information from sensory glands, muscles, and inner organs to the spinal cord and the brain. We also have what are called motor neurons, which transmit information from the central nervous system to muscles and glands. And last, we have our interneurons, which transmit information from neuron to neuron. These are going to be the most common type of neurons that we have in our nervous system because there needs to be a lot of communication that exists between our neurons to be able to have them reach the brain and the area of the brain for interpretation so that a proper response is going to be made by the individual. Now, in terms of our neurons, we also have what are called glial cells. And there are other cells in the nervous system that support neurons by moving waste products and provide insulation to the axon and assist neurons in communicating with other neurons. And now I also mentioned we do have what's called our myelin sheath, which is this protective insulation that's going to cover the axon of certain neurons and help speed the transmission of the nerve impulses. So again, taking that information through those electrical impulses. And we also have what are called the nodes of Ranvier. Now, the nodes of Ranvier are the gaps that exist in the myelin sheath that create non-insulated areas along the axons. So what we typically tend to find is that when multiple sclerosis occurs in individuals, this is ultimately a disease of the central nervous system in which the myelin sheath is going to be damaged or destroyed. So whenever it is that you think of individuals that say, well, that they have multiple sclerosis, or they have these kind of jerky movements, or their body isn't able to communicate and process information the way that they want it to process. So in other words, I want my arm or my leg to move, but it's not moving. Well, how does that happen? Possibly because of nerve damage, or also because you may have damage to that myelin sheath that's either damaged or destroyed. So as I was mentioning, the cell body does something that's very, very important. And what it does is it actually is ultimately going to be able to create an action potential if needed. Now, when information is coming from one neuron to the next, that next neuron is currently in what we call a resting potential state. This is where the electrical potential across the cell membrane of the neuron is in a resting state. So in other words, there's no activation at this moment of that neuron to tell it to fire and to continue to carry that information through. So when the cell now becomes stimulated, sodium gates at the base of the axon open. Positively charged sodium ions rush into the cell. These then cause the area inside the cell membrane at the point of excitation to become less negatively charged and become what we call depolarized. Once depolarization occurs, an action potential can exist, and this is where the abrupt change from a negative to a positive charge occurs, and a nerve cell now creates that nerve impulse. So in other words, now we have activation of that neuron and continued firing of that neuron to be able to carry that information through. Because remember, the purpose of the neuron is to carry information through from the perceptual site, right? Let's say, for instance, your eyes, all the way to the brain for interpretation. So as it's going through all these neurons, they got to continue to fire one after the other after the other. The only way that happens is if they depolarize. So in other words, they change their state from a negative state to a positive state. and ultimately an action potential occurs. So when that change occurs, because since the neuron's not active, right, it's not firing, now the neuron becomes active. When that energy changes, it's going to do the following. On the left-hand side here, we have our negative state, right? So we have our resting potential state. But now we're going to have our action potential occur. What does it mean for the action potential to occur? Again, we get depolarized. So in other words, there's that drastic change that goes from a negative state to a positive state. And now there's that rush of energy and that rush of positive charge to ultimately create that nerve impulse and be able to carry that information through. Now, as that information is getting carried through and it's going through our axon, now the information is being carried out, right? The information is being carried out. It gets to your terminal buttons. What now happens at the terminal button site, right? So what happens whenever it is that we get that information at the terminal button and there is that gap between one neuron to the next? Again, our synaptic cleft, right? Or our synaptic gap. So what's going to happen here? Once that information it reaches that terminal button, we're going to have neurotransmitters being released. What are neurotransmitters? Neurotransmitters are chemical messengers that are going to be released by the terminal buttons of the axon at the arrival of that nerve impulse. And these carry the message across the synapse to the next receptor site of that next neuron. Now, neurotransmitters have to fit into those receptor sites for that next neuron to capture that information and to continue to take that information through to say, you need to continue to fire. If not, if those neurotransmitters get released and that receptor site does not capture them because they don't fit, right? So we have to imagine them kind of like a key and lock mechanism. The neurotransmitter is the key. Those get released by the terminal button. The receptor site is the lock. The key has to be able to fit into the lock. If it doesn't, you're never going to be able to open that channel, right? You're never going to be able to open that lock. So what happens in this particular case? We now then see that nothing is going to occur. That next neuron will not fire if that receptor site does not take that neurotransmitter in. If it does and they fit, now that next neuron is going to be able to say, yes, I'm going to fire because I got the information to continue to fire. Now, we find that sometimes drugs block the action of neurotransmitters by occupying receptor sites, and we call these types of drugs antagonists. So what are antagonists? Think of, for instance, pain medication. Pain medication ultimately is an antagonist. And why? Well, because when you take that pain medication, you are essentially telling the neuron, don't allow that message of pain to go from point A to point B. So ultimately here, that medication, that drug is blocking those receptor sites on the next neuron at those receptor sites, and those neurotransmitters cannot get into the receptor sites. So that key is getting released by the previous neuron from where pain is originating, but that next neurotransmitter, that lock is being blocked. And it's being blocked by that drug, right? That's that function of the antagonist, to be able to block that receptor site so that way the message doesn't continue through. And you say, wow, I feel better. I don't have any pain anymore. Once that drug wears off, what's the next thing that can potentially happen? Pain comes back. You say, oh, now I got the pain again. Well, yeah, because the drug wore off, they get released, right? They no longer occupy the receptor sites of that neuron, and now those neurotransmitters are still being released by that previous terminal button, and now the key is fitting into the lock. And now it's saying, yep, there's pain again. Send the message through that there is pain. So we have lots of different types of drugs, a lot of different types of neurotransmitters that also get released in our brain whenever it is that we go through different experiences. And one of those most important ones is what we call dopamine. Dopamine is a neurotransmitter that's involved in movement and processes of learning and memory, feelings of pleasure or well-being, and also social bonding. We do find that sometimes there are irregularities in how the brain uses dopamine, and that may actually help explain the development of schizophrenia. What is schizophrenia? A severe and chronic mental disorder that's going to be characterized by disturbances in thinking, perception, emotions, and behavior. So sometimes the brain releases dopamine incorrectly or doesn't release it at all, thus creating some of these effects. Now, for Parkinson's patients, those individuals have a degenerative brain disease that involves the destruction of dopamine producing neurons. And it's characterized by muscle tremors, shakiness, rigidity, and a difficulty in walking and controlling fine body motor movements. Now, why does that happen? That happens because information isn't being sent through to say, don't have these movements, or have these movements inappropriately in these types of situations. Now, as we were talking about different types of neurotransmitters, we were talking about antagonists. Remember, antagonists are going to be the drugs that either increase the availability or effectiveness of neurotransmitters or mimic their action when binding to the receptor sites. We also can have mild stimulants such as caffeine, which increases the availability of the neurotransmitter glutamate. And we also have amphetamines like cocaine and stronger stimulants that are agonists that increase the availability of dopamine by blocking its reuptake. So we tend to find, again, that there's a lot of different factors and a lot of different drugs that can potentially have different effects. Alcohol and anti-anxiety drugs act as agonists by increasing the sensitivity of receptor sites to gamma aminobutyric acid, the majority inhibitory neurotransmitter that's in our brain. Our antidepressants are going to be drugs that are going to be used to treat depression and work as agonists to increase the availability of the neurotransmitters norepinephrine and serotonin. So norepinephrines are inhibitory neurotransmitters that have pain-killing and pleasure-inducing effects. And again, whenever it is that we feel pain, this is what ultimately is going to happen. Think about athletes. They get hurt while they're out on the field, but yet they still play through the pain. Sometimes they don't even recognize that they are in pain, that they're cut, that they got a broken finger or a broken foot, and they continue to play. How do they do that? Well, because during that time of them being engaged in that activity, endorphins are being produced. And as those endorphins are being produced, they're having that pain-killing or pleasure-inducing effect. Once it is that they get out of the game and they go and they sit down, now all of a sudden we start realizing that they start having a lot of pain. Now, as we're looking and talking about our nervous system, let's take a closer look at how our nervous system is actually organized. Now, there's two major components to our nervous system. Those two components are our central nervous system and our peripheral nervous system. The central nervous system is the body's master control unit. This includes the spinal cord and the brain. The peripheral nervous system is the body's link to the outside world, and this is made up of our autonomic nervous system, the somatic nervous system, and the autonomic nervous system is made up of two subsystems called the sympathetic and the parasympathetic nervous system. Now we're going to talk a little bit about both of those in just a minute. Looking more closely at each one of our central nervous system components, we're going to be able to identify with the central nervous system. As I mentioned, this is the body's master control unit. So within the central nervous system, we have the spinal cord and the brain. The spinal cord is going to be a column of nerves between the brain and the peripheral nervous system that allows for communication, while the brain is going to be divided into three major parts that we're going to see here on our next slide. Those three major parts are going to be the hindbrain, the midbrain, and the forebrain. Now, in terms of our peripheral nervous system that's made up of our autonomic nervous system, which regulates involuntary bodily processes, including your heart rate, respiration, digestion, and pupil contraction, it also operates automatically without conscious direction. And the autonomic nervous system is made up of two systems, the sympathetic and the parasympathetic nervous system. The sympathetic nervous system mobilizes body resources in response to threats by speeding up the heart rate and respiration and drawing stored energy from bodily reserves. This is where we talk about the fight or flight syndrome, right? Or the fight or flight response, where whenever it is that you're encountering a very stressful situation, you either stay and fight or you flee. Either way, your sympathetic nervous system is going to become activated because our bodily resources are going to be used in response to that threat. versus your parasympathetic nervous system, which replenishes bodily resources by promoting digestion and slowing down other bodily processes. This is normally where we're at whenever it is that we are resting. We are in a parasympathetic nervous system response. Now, the other part of the peripheral nervous system is going to be the somatic nervous system. Now, in terms of the somatic nervous system, this carries sensory information from sensory organs to the central nervous system and relays motor movement commands to muscles and controls voluntary movements. So our somatic nervous system is going to be very important to carry that information from when it is that we talk about carrying information from our specific sensory organs like our eyes, our ears, our nose, our mouth, and our touch senses, and being able to process that information. Now, as I was mentioning, there are 3 divisions of the brain. The 3 divisions of the brain are the following: the forebrain, the midbrain, and the hindbrain. So let's begin first with the hindbrain. What is the purpose of the hindbrain? The hindbrain is the lowest and, in evolutionary terms, the oldest part of the brain. The hindbrain includes the medulla, the pons, and the cerebellum. The medulla is involved in regulating basic life functionings such as your heartbeat and your breathing. The pons is involved in regulating states of wakefulness and sleep, while the cerebellum is involved in controlling and coordinating and balancing. Now, if we move over to our midbrain contains pathways that connect the hindbrain and the forebrain. Parts of the midbrain make up what we call the brainstem or the stalk in the lower parts of the brain that connects the spinal cord to higher regions of the brain. Now, the midbrain includes reticular formation, which is going to be involved in regulating states of attention, alertness, and also in arousal. Now, our last part of our brain the last division of our brain is going to be the forebrain. Now, the forebrain is the largest and uppermost part of the brain. This includes the thalamus, the basal ganglia, the hypothalamus, and the limbic system. Now, the purpose of the thalamus, it serves as a relay station for sensory information and plays a key role in regulating states of wakefulness and sleep. The basal ganglia is a cluster of nerve cells that are involved in regulating voluntary movement. The hypothalamus helps regulate many vital bodily functions, including body temperature and also emotional states and responses to stress, while the limbic system plays an important role in memory and emotional processing. Now, aside from those 3 divisions of the brain, we also have a number of lobes in the brain, which we actually have 4. Now, the 4 lobes of the brain are going to be our frontal lobe, our parietal lobe, our occipital lobe, and our temporal lobe. Now, for our frontal lobe, we handle executive functionings. So the frontal lobe, which is in the front part of the brain, this is going to be able to handle those executive functionings, and we call it the decider. It plans movements, is involved in memory, and inhibits inappropriate behaviors. Now, we do find that sometimes with individuals who have frontal lobe damage, we find that the frontal lobe isn't working. Who suffer from frontal lobe damage, we find that they tend to act before they think. Now, instead of the frontal lobe being able to inhibit inappropriate behaviors, we find that individuals with frontal lobe syndrome tend to engage in behaviors of what we would call risky. So they may engage in addictive behaviors such as gambling, where they can't stop themselves from gambling or engaging in other risky behaviors. Now, our second lobe that we're going to discuss is what we call our parietal lobe. Now, our parietal lobe is going to be responsible for monitoring body sensations such as pain and temperature. Now, our third lobe of the brain is going to be our temporal lobe. Our temporal lobe is important for emotions and language processing. Now, later on in the semester, when we talk about language, we're going to talk about 2 very important areas, such as Broca's area and Wernicke's area. Now, we tend to find that individuals who suffer damage to either Broca's or Wernicke's or both areas tend to have different language processing ability issues. For those who engage or who have Broca's aphasia, right, who have a lesion or damage to Broca's area, have difficulty producing words. While individuals who suffer from Wernicke's aphasia, they have damage or lesions to Wernicke's area tend to have issues with comprehending information. Now, we also find that individuals may suffer from what's called prosopagnosia, where they have an inability to recognize faces whenever they may have excessive damage to the temporal lobe. Now, our last lobe that we're going to discuss here is our occipital lobe. Now, our occipital lobe is responsible for visual processing. Now, in terms of visual processing, again, this is also going to be correlated with being able to recognize faces, memory, and a lot of other visual processing issues that are going to be very typical for the processing in the occipital lobe. Now, whenever it is that we are trying to assess individuals' brain functioning, so how is it that the brain actually works, we can also use a number of different techniques. Now, there are 4 techniques that we're going to discuss here. These include what we call PET scans and CAT scans. PET scans are what we call positron emission tomography, and these show activity of the brain by ingesting a radioactive tracer, and we're able to label those specific areas of the brain that are active while they're engaging in that specific behavior or action. Now, CAT scans, on the other hand, which are computerized axial tomography, only show the structure but do not show any type of activity. What we would then label as our modern day phrenology, where we would say, here are the different structures of the brain, maybe this is what they do, but we can't really see any type of activity while the individual is engaged in the activity itself. Now, the final 2 techniques that we're going to talk about are going to be our MRI, which are our magnetic resonance imaging, which assesses soft tissue activity, and this is measured through size. So as blood flow goes to those specific areas, we tend to find that they become a little bit larger as more blood is being pumped into those areas of the brain, thus indicating for us that there is activity in that area. Versus an fMRI, which is our functional magnetic resonance imaging technique, which measures blood flow and oxygenation in the brain using iron in this big magnet that we put you in. So as more blood flows to those areas, blood contains iron, and that magnet is able to identify where there is that iron in the blood, more blood flow to those areas, thus indicating activity in those regions. All right, that wraps up our discussion associated with neuropsychology. I hope everyone enjoyed today's lecture. If you do have any questions, please do let me know.